Switching to something other than plants, I'll be talking about old world fruit flies. So what is a fruit fly? When normally people think of fruit flies, they think of Drosophilidae. But I'll be talking about true fruit flies in the family Tephrididae, which actually attack fruits that are still in the plant, and then the larvae cause direct damage to these fruits. So this is a group that's highly economically important. This tribe, Vicini, is native to the old world tropics. There's over 800 species, 100 of which are economically important, and many are highly polyphagous. Especially this one, Bactricera dorsalis, or the oriental fruit fly. And you can see this peach has been attacked by fruit flies, and you can see the damage it causes. There are 4 genera, but the main ones I'll be talking about are the genus Dacus and Bactricera, with 500 and 200 species, respectively. So the native range of Bactricera is South Asia, Southeast Asia, Australasia into the Pacific, and the native range of Dacus is South Asia and the subtropical and tropical Africa. But these guys have spread around the world through agricultural trade. There have been some previous phylogenetic studies looking into this group. The first large Dasein phylogeny was Croce et al. 2013. Used 3 genes, a lot of missing data, a lot of data from GenBank, so a lot of the results are kind of iffy at best. And then Virgilio, 2015, focused mainly on Ceratitis, which is a sister group, but included some Dacini flies. And the main results from both of these studies was that Dacus falls within Bactricera, rendering it paraphyletic. But since they used there was a lot of missing data in both these studies, this result was kind of not well supported. Croce et al. Also did some biogeographic analyses, and this is the scenario that they came up with their phylogeny. Bactricera and Dacus are evolved on India subcontinent around 80 million years ago, and then Dacus dispersed into Africa. Then there's some back dispersals back into India while it was still in the Indian Ocean. And then once India rose into and hit Southeast Asia, the groups spread both ways to Southeast Asia, and then some back dispersals from Southeast Asia and Africa. Dasein flies also have some interesting traits. There's high species diversity despite low morphological variation. You can see on your left here, the melon fly kind of looks like a wasp. It has the causal band on the wing that mimics the way the wing folds on a wasp, and then the coloration as well. It's not a perfect mimic, but it seems like it's good enough that there's a selective pressure to keep this wasp form, which limits the amount of morphological characters that separate species. And thus, a lot of these species groups based on morphology have been put into question. And this is just a dorsal view of a female papaya fruit fly next to another wasp. You see how the ovipositor kind of looks like the wasp's stinger. So any predator that sees these flies might be taken aback and worried about being attacked by the wasp. And another interesting aspect is this male kairomone response. So males respond to chemicals from plants, and this trait has been used as one of the main methods of controlling these species. You put these kairomones into traps and reduce the populations of males in agricultural environments, which basically reduces the populations and saves your plants, or it saves your crops. But it's also been used to identify morphologically similar species, because you can have 2 species that look exactly the same, but one responds to one kairomone, another responds to another, and you can say that they're different. And these are the 3 known kairomones: raspberry ketone, methylugenol, and zindrone. Raspberry ketone and methylugenol have been studied for many, many years, over 50 years. It's been well documented. Raspberry zindrone has been recently found within the last 5 years, so there's still kind of questions of what it does and how many species actually respond to it. And you can see how different groups have different, use different lures, or use different kairomones. Dacus is primarily only raspberry ketone. Zoogodactylus as well, but Bactricera has a more 50-50 split, 65-35 fracture. And our study is the first to use syndrome responding flies to look at their phylogeny and where they place out. So the reason kairomone response is kind of interesting is that the males ingest these kairomones and incorporate it to the male mating pheromones. So females actually really like males who have ingested these chemicals. This actually induces physiological changes in the male flies. They are more active, they have different mating dance, they actually die faster because they're so active. And it's been theorized this ingestion of these chemicals is part of a disruptive selection mechanism where if you have an incipient species, if they actually switch to a different kairomone, the males ingest a different kairomone and the females like it, it effectively isolates that species from its sympatric populations. Which may be a component of driving diversification in this group. So some of the questions we want to talk about are how are Dacus and Bactricera related? In previous studies, Dacus rendered Bactricera paraphyletic, but with low support. There's a more robust phylogeny. More robust phylogeny say the same thing. Our groups based on morphology supported in this phylogeny, and then is our phylogeny and dating congruent with previous studies. And then once we've dated our phylogeny, we use that to run some biogeographic analyses and then look at how kairomone use has evolved throughout this group. So we collected specimens mainly in Southeast Asia, in Australasia, and then we were able to obtain specimens through collaborators throughout their native range as well, and invasive ranges. We sequenced 165 species out of the 800 for 9 genes, 1 mitochondrial, to 8 nuclear. And we did standard phylogenetic analyses, Braxamel, booster days, these for the divergence time dating, Biogeography pairs for the biogeographic analysis, and then SynMap to look at evolution of kairomone response. And this is the tree that we got. And basically, in this slide, I just want to show that the group Dacini, the tribe, is well supported in both Bayesian and maximum likelihood analyses. And then also, Dacus is rendered paraphyletic with regards to Bactricera. Bactricera is rendered paraphyletic with regards to Dacus as well with high support. So with a more robust phylogeny, that relationship is well supported. When we look at these morphologically identified subgenera, we find that many of them are paraphyletic as well, or render other genera, subgenera paraphyletic. So this just kind of shows that this whole group is kind of a mess, and we need to do massive revisions to the taxonomy of this group. Then we wanted to date this phylogeny as well, but there wasn't any in-group fossils that we could use, so we had to use out-group fossils based on the Nematopsera Bactricera split, Bactricera crown fossil, and Schizophora crown fossil. So these are pretty distantly related, or pretty distant fossils. And this is our basic phylogeny, or dated phylogeny, showing the mean divergence times of the major groups. When we compared this to the Crochet et al. Paper in 2013, we find that many of our mean divergence times are actually younger than what Crochet et al. Came up with in their study. And we think this is because of more comprehensive gene sampling and also inclusion of more nuclear genes as well. Because we use pretty much basically the same fossil calibration models. So we wanted to use this phylogeny, or the stated phylogeny, and then look at biogeographic scenarios. So we took all our species and then did put them in different biogeographic zones corresponding to where the ranges are. And I won't be showing the output from biogeography, but because it's a little complicated. But here's the basic scenario. We have the sea mean dispersing into the Indian subcontinent from Africa around 50 million years ago. There it diversified to form all the major subgroups and subgenera and genera that we find. Once India ramps into South Asia, the groups spread out. Dacus going both into Africa and Southeast Asia, where Zeuglodacus and Bactricera go to Southeast Asia. And once they got into Southeast Asia and Australia, they spread even more widely and diversified greatly there. So that was kind of the basic biogeographic hypothesis that we came up with our phylogeny. And we wanted to use this phylogeny also to look at the evolution of kairomone response, since it may play a role in driving the speciation of this group. So blue indicates species that respond to method of regional. Red is raspberry ketone, green is dendrone, and Na is doesn't have any kairomone response. And what we find is, especially in the subgenus Bactricera, kairomone response seems to be a readily switchable trait, where you can go from red to blue to back to red again, or from red to green and back and forth. So this seems like that this kairomone response is a plastic trait that this could be just the first line of evidence suggesting that this response might be a driving component in the diversification, and we have to look into it further, studying more the species more closely than in phylogeny. So in conclusion, Bactricera and Dacus are not sister genera. Dacus renders Bactricera paraphyletic. Many of the species and subgeneric groups are paraphyletic as well. The biogeographic hypothesis is slightly different from the previous studies. There's not this back and forth between Africa and India, but it seems like our hypothesis seems like more parsimonious, considering the plate tectonic movements. And kairomone switching is prevalent in this group, especially in the subgenus Bactricera. So I just want to thank my community members and everyone who's helped me collect species and all this collective work, and also all these governments who help permits to collect.